So as I said, we have Anna with us this week. She's a senior lecturer at Edith Cowan University, and she specializes in plant, fungal, and fauna interactions, both in broadleaf and in conifer forests. And she's worked all over the world. It was really cool to see where Anna has been. She's been both here in Australia, New Zealand, as well as all the way out to Sweden. And she's published some really cool work looking at that interaction between how foragers interact with fungi and how they might be spreading fungi. And, because of work like this, like Anna's work and others' work in this area, whenever I go into a forest now and I see, these little holes dug, I'm really curious to know what used to be there or what is there. And I'll just end with one fun fact that I, really want to know more about one day. Anna has a diploma in modern languages. So not just looking at the fungi. So I'm curious to see how that fit in with, your idea of where your career was going. So with that, I will be quiet and I'll pass it over to you, Anna.
Thanks, Jonathan, for the lovely introduction. Kaya, everyone. Thanks for coming along to hear this seminar today. So I just wanted to start by acknowledging that I'm coming to you from Whadjuk Noongar country and pay my respects to the elders past and present, the Whadjuk Noongar people, and also pay my respects to the Pinjarra people who are the custodians of the lands that I've done a lot of this research on. So what I wanted to do today was just to share with you a couple of different studies that we've been doing over the last 5 to 10 years, looking at how soil fungi respond to disturbances, particularly soil fungi in the southwest of Western Australia where I'm based, but we have extended some of this work out into other areas. So I'm sure you're very familiar with the fact that soil fungi play these really key roles in ecosystem resilience and resistance. So things like mycorrhizal fungi are clearly very important for the growth of plants and for obtaining nutrients and water for those plants from the soil, and other things like wood decay fungi or leaf litter decayers, saprotrophs in our system are really important for nutrient cycling. So they're clearly really important for the way that ecosystems function, and that's one of the reasons that I'm really interested in working with soil fungi. And one of the things that I've been working on in the last little while is how disturbances might change the communities of fungi that are present in the soil and how that might then lead to changes in the resilience and resistance of those ecosystems that we're operating in. So disturbances can impact soil fungi in a range of different ways. They can impact them directly by changing the structure of the soil, by changing the nutrients that are available in the soil, and by changing the other types of substrates, so types of woody materials, for example, that are present in the soil. They can also operate on soil fungi indirectly by changing the composition of the plants or the animals that are present in those ecosystems, and therefore changing the, say, different plant habitats that are available for those fungi in the soil to survive on. And because the fungi are so well integrated into this system, structural shifts in soil fungal communities can have a really close relationship with changes in ecosystem processes. So they can in some ways be a kind of a proxy, I guess, for those changes in ecosystem processes and functions. So I want to talk to you today about 2 different case studies that we've been doing, and they're kind of collective case studies, I suppose, over a range of different studies that we've done. And the first one is looking at how communities of soil fungi respond to drought and fire. And this is based on a number of different studies that we've done in the southwest of Western Australia, in the Jarrah forest. So it's one of our dominant forest ecosystems just outside of Perth. None of this work that I do happens in isolation, so I want to acknowledge the co-authors that I've been working with from Murdoch University and from the Department of Biodiversity, Conservation and Attractions here in Western Australia, and from Edith Cowan University as well. And so first I just want to talk a little bit about the background to our thinking in these studies. So I'm sure everyone's really well aware of the fact that our climate is changing, and as a result, we are having hotter droughts, so droughts and heat waves, and they've been reported from almost every forested continent throughout the world. Fire is also a really current contentious subject, and fires have also been increasing in incidence and severity. And there are some amazing examples throughout Australia of how this has been happening in the last 5 years or so. And so what we were really interested in was trying to understand how these changes in disturbance regimes, so increasing hotter and drier conditions, but also increasing hotter and potentially more common fires might change the soil fungi, and how that might then lead to some changes in ecosystem processes. So in the southwest of Western Australia, we've had a protracted drying period since about the 1970s. You can see in this figure on the right that the weather has been drying over the last 50 or so years. But in 2010, we had a particularly dry winter. It was one of the driest winters we had on record to that point, with about 40 to 50% below average rainfall. And this was then followed by a heat wave in the summer of 2011. A heat wave is defined, I think, as anything over more than 3 days over 35 degrees. And in this particular year, we had 9 days over 35 degrees. So it was quite a significant event. And following this period, my colleagues at Murdoch University, particularly George Matusik and Katinka Ruthroff, noticed some widespread tree die events throughout the Jarrah forest, just east and south of Perth. So on the left here, you can see a figure, and the dark shaded areas are basically the extent of the northern Jarrah forest outside of Perth. And on the right, you can see some aerial imagery of those die-off patches. So in those die-off areas, they represented about 1.6% of the northern Jarrah forest, so quite a significant area still, about 16,000 hectares. And they were in small isolated patches where the trees had just completely died. Those dominant trees being mostly Jarrah and Marri. George and Katinka did a lot of work looking at how these, why these particular areas had died off, and found that they were concentrated in patches where there were shallower soils and they had a much lower water holding capacity. So often in the proximity of granite outcrops and things like that. So there's been quite a lot of work on the impact of these drought sites, we've called them. And there's been some concerted efforts coming out of Murdoch University over the last 10 years trying to understand what the impact of that has been on the ecological communities, particularly the tree and understory communities, also the animal communities, all those kind of things in response to this drought. But what we were really interested in is what had happened to the soil fungi in these areas. The dominant tree species, Jarrah and Marri, are both mycorrhizal species, and so we were particularly interested to see whether those significant die-offs had any impact on the fungi in the rhizosphere of those trees. This is just a little bit of data that shows how those ecosystems had changed in response to the drought. So over the next couple of slides, I'm going to show you some figures where we looked at areas that had been droughted. So they'll be on the left of the figure, those that have been minimally impacted by the drought, and those that have been seemingly unaffected by the drought. And so you can see, for example, that the number of stems per hectare of those dominant tree species had changed quite significantly in response to the drought sites. You can also see that the canopy cover had reduced quite significantly in the droughted sites. So because most of those trees had died off, the canopy cover had reduced quite significantly. And you can also see that the ground cover had changed quite a lot. So there was a lot more, there was much less leaf litter in those drought sites because of it all, because there was not so much canopy cover dropping leaf litter in those areas. So all those things could have an impact on our soil fungi, and we're really interested to see what might happen in that particular case. So we collected soil from the rhizosphere of Jarrah trees in the drought sites and in associated sites. So we looked at what we called a drought impact gradient. So we collected from areas that were severely droughted, adjacent areas about 50 to 100 metres away that were minimally droughted, and then we moved again another 100 metres or so away to areas that were not impacted by the drought or didn't have significant tree die-off. So we collected the soil from the rhizosphere of these jarrah trees and subjected it to high throughput sequencing. We sequenced the ITS2 for those of you who are interested in that kind of thing, to try and look at what the different fungal species were in that soil. And we looked at fungal species richness, diversity and composition, and particularly we wanted to understand whether there are any changes in key fungal functional groups. We didn't find any significant difference in the number of fungal species in the under the different treatments. So you can see on the left here, this is both richness and diversity. And you can see on the left here, we've got our drought sites. In our drought sites, we still had a significant number of dead trees. So we collected from dead trees, which is shown in the red, and live trees that are shown in the blue. And you can see that there is a monotonic decrease in species richness and diversity with decreasing disturbance, but that wasn't significant, but there was a bit of a pattern there all the same. When we looked at the community composition of those species in the drought sites, though, we saw a really clear pattern. So our drought sites in this figure are the orange ones, the minimally affected ones are the green ones, and then the unaffected controls are the blue ones. And so you can see there's quite a shift in community composition of those soil fungi as you're moving away from those drought sites. So that was really interesting, and we drilled down into that a little bit further and looked at how some of those different functional groups were changing. And we saw some really interesting patterns. So these are some of the different functional groups that we looked at. We determined function by linking it to the taxa that we were able to identify from our sequence reads. And what we found was that there was a significant decrease in the number of ectomycorrhizal fungal species in those droughted sites. And that there was an increase in the number of pathogens, for example, in the bottom right-hand corner, particularly with those dead trees. And that inverse to the decrease in ectomycorrhizal species, there was an increase in ericoid mycorrhizal species and buscular mycorrhizal species. So these were some really interesting patterns that we saw, and leads to some really interesting questions about about the resilience of those forests over time, if they're losing some of those key ectomycorrhizal species that might be helping in seedling recruitment or in maintaining the health and fitness of those adult trees over time. So this is an example of what happens when you have just one disturbance, but we were also, I don't know if lucky is quite the right word, we were fortunate enough to be able to look at the impact of two or overlaying disturbances as well. So we also looked at the impact of drought followed by a severe wildfire in these forests. So in 2016, 4 years after the drought occurred, part of the droughted area was also subject to a severe wildfire. And those of you who are in Western Australia might remember the Waroona Yalloop fire. It was quite a widespread fire and very, very high intensity. And so we were interested to see whether that compounding effect of wildfire and drought might have a different effect on the soil fungi that were present in these systems. So we collected samples again from the rhizosphere of the dominant tree species, and those samples were collected 4 years after the fire, but 8 years after the drought. And we collected them from an array of different sites, either high drought or low drought, and then high fire or areas that were not burnt. So again, we collected rhizosphere under the soil of just living trees in this case, and subjected them to sequencing for fungi, sequencing the ITS2. And this time we also did bacterial sequencing too, because we wanted to see whether there was any difference in how the bacteria responded to these different scenarios. But today I'll just present the fungal results because I figured that's more interesting for everyone here. So what we did find was that, so we've got, in this figure, this is the species richness of fungi. We've got fire, as the 2 bars on the left, and unburnt sites are the 2 bars on the right. And then the pink bars are high drought intensity and the blue bars are low drought intensity. So what you can see is that there's a decrease in the number of ASVs or species of fungi with increased disturbance. So the more disturbance you have, the fewer species of fungi you're finding in that soil. When you look at how that impacts the community composition of those fungi, you can again see that there's some quite significant differences between those areas depending on what disturbance types they've had. So we've got high fire, high drought in the orange here, high fire severity, low drought in the green. The purple is out control, so unburnt and low drought, and the blue is unburnt and high drought. So you can see there's quite a bit of difference between those different treatments. And so they're all having some impact on the way that the fungi are responding. When we drill down again to look at how some of those different key functional groups were responding, what we could see again was that the ectomycorrhizal species in the middle at the top there were responding in a similar way to the overall species richness. So there was a decrease in the number of ectomycorrhizal taxa with increasing disturbance. And the same in this case with the ericoid mycorrhizal species, there was also a decrease in the number of species. And this is really interesting because this is still 8 years after that initial disturbance of drought and 4 years after the fire. So you can still see some quite clear differences in those fungal communities, even though it's quite some time since the disturbance occurred. You can also see a decrease in the plant pathogens with increasing disturbance and in those wood saprotrophs. But with the litter saprotrophs, we didn't really see much difference. We didn't find a huge number of arbuscular mycorrhizal fungi, but we did find that there were quite a lot of arbuscular mycorrhizal fungi where there was high drought but no fire. So that was interesting as well. So what we're seeing here really is quite a significant change in some of these functional groups. And I think that it'll be really interesting to tease out some of these differences and see whether they really do translate into, say, changes in recruitment patterns within these forests or changes in the capacity of the forests to then resist further disturbance, or whether they mean that the trees are no longer as resilient as they used to be because they don't have some of these key fungal partners anymore. These kind of patterns are being seen in lots and lots of areas. We've known for a long time, for example, that there are some fungi that do respond positively to fire and will only fruit after fire, or that will only fruit in long unburnt areas. So there's quite a lot of information about that. And some recent work by Hans Amitsbol and Sue Baker in Tasmania has also shown similar patterns to what we're showing, that there's a decrease in fungal diversity and decrease in fungal species richness with increasing disturbance of fire, and that there's a decrease in the mycorrhizal species in response to fire too. So that on the right-hand side, that's those green coloured bars, the symbiotrophs there. So looking at the impact of compound disturbances on our soil fungi really does lead to some very important questions about what changes these will have on the resistance and the resilience of ecosystems over time. We're likely to have more and more of these kinds of disturbances in the future. And if we keep having stacked disturbances in this way, it will be really interesting to see how this relates to the capacity of the forest to regenerate after each, subsequent drought event, how it might lead to changes in the forest's ability to cope with chronic changes in temperature and rainfall, and how the regeneration after fire might occur in the future. So it's a bit of a gloomy story, I suppose, in some ways, but some really interesting things to continue to investigate in this space, particularly, I think, in the way that seedlings might respond to these particular changes in soil fungal communities and whether that will impact regeneration in that space. Okay, so the second case study that I wanted to talk about was the work that Jonathan mentioned before, looking at digging mammals and how they might impact soil fungi. So we've got one of our cute little quenda here. It's related to the southern brown bandicoot. And again, this work never happens in isolation. I work with a whole lot of wildlife ecologists and other forest ecologists on these kind of projects. So I've got strong collaborations with UWA and Murdoch and Department of Biodiversity, Conservation and Attractions here in WA. And I've just pulled out some results from a range of different studies that we've been doing in this area over the last 5 to 10 years to give a kind of overview of the information that we're obtaining. So if you're not familiar with digging mammals are widespread throughout Australia. They're things like potoroos, bettongs, wombats, anything that digs in the ground for to look for food resources. So it might be digging for underground fungi or roots or bulbs, or anything that burrows underground like wombats or bilbies. So throughout Australia, there's been a significant decline in the number of digging mammals that we have. So you can see on the right-hand side here, digging mammals used to be quite widespread. So that's the green bars showing how widespread some of the digging mammals used to be. And the black bars are how widespread they are now. So you can see, for example, if you look down at the Woylie, it used to cover about 40% of Australia's landmass and distribution, and now it's a very, very small proportion. The map on the left shows that further. So that's a little Woylie there. And you can see that its distribution used to be in that grey area all throughout Australia, but it's now just found in this tiny little area of black in the southwest of Western Australia. These digging mammals are really important. One of the reasons that they've declined over time is because a lot of them fall within the critical weight range, which are the animals that are easily predated by cats and foxes, but it's also as a result of land clearing and other human-related disturbances. The one digging mammal that hasn't really been affected by these kind of disturbances is the echidna, and you can imagine it's not a very good meal for a cat or a fox, and they seem to persist throughout Australia quite comfortably still. Digging mammals are what we call ecosystem engineers, and there's been a lot of work to understand what their impact might be in ecosystems and what the impact of this kind of digging activity might be. Particularly my colleagues Leonie Valentine and Trish Fleming have done a lot of work in this area. And what they have found is that the digging activity, even of something like a quenda or a woylie, a bandicoot or a bettong that just digs in the ground to look for food, can have a significant impact on the ecosystems that they live in. So a quenda, for example, can turn over almost 4 tonnes of soil in a year. So even though it's just doing lots and lots of little digs to find food, it's turning over a significant amount of soil at the same time. The diggings that they create look a bit like this on the right-hand side. So you can see that there's the pit where they've been digging in and probably sticking their nose in to find something delicious in the bottom of the pit. And then there's the spoil heap, which is the pile of soil that they put behind them as they're digging. And this action of soil turnover has some really clear implications for the structure of the soil and some other ecosystem contexts. So it can increase the soil heterogeneity. It changes some of the mechanical properties of the soil. So it can change the texture and the structure and the density of the soil. And it also often helps to bring some of those nutrients to the surface and mix those nutrients around. This is an example of what a dig looks like. So this is a woylie dig from Karakamara in Western Australia. And you can see quite clearly that it's got the little area where it's dug, but it's also got that spoil heap behind it. And that spoil heap is very importantly covering a whole lot of the leaf litter that was on the surface of the soil. This covering of the leaf litter is really important because it's helping to incorporate a lot of that organic matter into the soil and lock that debris into the soil. It keeps that organic matter much moister and helps it break down much faster. So it increases the amount of organic matter that's being incorporated into the soil. Organic matter can also accumulate in the pit as well and break down much faster because it's nice and moist in there. So it's increasing the amount of organic matter that's in the soil, which increases soil health and the capacity for soil biota to persist in the soil. It also increases nutrient cycling. And there's been some really great recent work by Leonie Valentine showing that it can also lead to cooler fires because a lot of those surface fuels are being incorporated into the soil and so are not available for burning. As well as that, digging mammals can increase water infiltration. The pit itself can break the hydrophobic layer on the surface of the soil that's really common in a lot of our Australian ecosystems, and allow that moisture to infiltrate into the soil much more easily. And as well as that, there's also been some great work that shows that those pits can also act as nice seed beds for seed retention and plant recruitment. So that increased moisture in those pits and also the kind of lower increased organic matter means that they're great areas for seedling recruitment too. As well as that, lots of these digging mammals are known to be mycophagous. Some of them are obligate fungal feeders. Some of them are just opportunistic fungal feeders. But in feeding on those fungi, they're really important for dispersing those fungi. So this is another example of a little woylie here who's eating some kind of fungal fruit body that it's dug up from the ground, and it eats that and then poos the spores out in its poo. And then by doing that, disperses them quite beautifully throughout the landscape. So for a lot of those hypergeous fungal species that produce hypergeous fruit bodies, so underground fruit bodies, this is one of the main ways that they are dispersed throughout the landscape. Lots of those specialized fungal feeders are really good at smelling out delicious truffles underground and really good at digging them up and then dispersing those spores. So it's, we like to think of this as a bit of a tripartite relationship between the digging mammals, the trees in the landscape, and the hypergeous fungi that live underground. Lots of those hypergeous fungi are mycorrhizal and form those specialized mycorrhizal associations with trees in the landscape. The trees provide important nutrients and food source for the digging mammals. The digging mammals also provide good habitat, good soil turnover and things that are important for tree growth. And then the digging mammals and the fungi have this special relationship in terms of food source and dispersal. So it's what we like to say is a tripartite relationship. So this is quite clearly established. But what we were really interested in was, if we are losing these digging mammals in the landscape, what's the impact of that going to be overall on the ecosystem health? And particularly if these digging mammals are really good dispersers of truffles, what happens when they're being lost in the landscape? So in Western Australia, one of the digging mammals that's still relatively widespread is the quenda. The quenda used to be part of the southern brown bandicoot complex, but has recently been recognised as its own species. And it's really quite widespread in urban areas in Western Australia. So if you've been to Murdoch University, for example, you can see them running around on campus, but you can find them in lots of areas as well. And lots of people are very familiar with quenda in Western Australia. Quenda are known to be mycophagous, but they're not obligately mycophagous. They're quite omnivorous. So they eat insects, they eat plants, they eat roots and corms and things that they dig up from underground. They eat termites and earthworms that they dig up from underground. But they also do eat fungi too. And so what we were interested in was trying to understand if we've lost some of these more obligate mycophagous species like woylies and potoroos from our landscapes, and they've essentially been replaced with quenda, which eat fungi, but maybe not so much. Are those quendas still playing a really important role in dispersing those fungi and dispersing fungi throughout the landscape? And are they adequately replacing those woylies and potoroos and things? So we collected quenda scat from some urban areas and native bushland in the south of Perth, in the city of Mandurah. And we analysed this to see what fungi were present in those scats. So we used the DNA and again sequenced the ITS2 region. And what we found was that there were lots and lots of fungi. Some quenda scats had up to 82 species of fungi in them, I think. And of course, those fungi won't all be species that are deliberately eaten. Some of them will be fungi that are present in other food or animal material that the quenda have eaten. But when we looked at the different functional groups of those fungi, we found that a significant number of them were actually ectomycorrhizal species that produce macroscopic fruit bodies that are likely to have been eaten deliberately by the quenda. So if you have a look at this figure here in the blue, you can see the ectomycorrhizal species. You can see that about half of the diet of these quenda was ectomycorrhizal fungi. And that was really good news because it means they are still theoretically dispersing them throughout the landscape. They also appear to eat a range of other species of fungi, whether deliberately or incidentally. And so they are acting as a vector for those other ecologically important species like saprotrophs and lichens and other important things. We were then interested to see whether those fungi that were being eaten were able to form mycorrhiza on tree seedlings. So what we did is that we set up a glasshouse experiment where we mixed quenda scat in with eucalypt seedlings and grew the seedlings for 3 or 4 months, and then harvested them and looked at the roots, extracted DNA to see whether some of those fungi that we were finding in the scats were also forming mycorrhiza on the seedlings of the eucalypts. And we had a number of different controls. We used autoclaved scats to control for the amount of nutrients that we're adding to the system. And then we also added spores from fruit bodies too, to see whether they were going to do a similar kind of thing. And in this figure, you can see in the top section here is where we added the scat to the seedlings. And about 30 to 40% of the fungi that were recovered from the roots of those seedlings were ectomycorrhizal species, and they were very similar to those species that we're finding in the quenda scat. So this was really great, a really great indication that the fungi that were being eaten by the quenda were able to form mycorrhiza on tree seedlings and then go on to improve the health of those seedlings and the ecosystems that were being found in. So it's really clear from this work that quenda, although they might not be obligately mycophagous, are still playing a really important role in maintaining ecosystem health. And that's really great news for us in Western Australia because they are still relatively widespread. So not only is their digging action really important, but also their dispersal of these mycorrhizal fungal species. The other thing that we were really interested in trying to understand was whether the digs themselves actually influence the soil fungal community at all. So whether that action of digging and incorporating more organic matter into the soil is actually going to change the fungal communities that we're finding within the soil. So we did an experiment where we went to 2 fenced reserves. These fenced reserves, one is just in the hills outside Perth and one's further south in the southwest of Western Australia. And they're both fenced reserves where they've reintroduced significant populations of digging mammals. So at Karakamaya, just outside of Perth, they've reintroduced woylies, which are an obligate mycophagous species, and the quenda that we were talking about before. And at Pera, down in the southwest, they have reintroduced woylies into these areas. So we collected soil cores from inside the enclosures and the forest adjacent to them outside the enclosures and grew jarrah seedlings in them for 3 or 4 months, and then had a look to see what different species we were finding on the roots of those seedlings. So we're using those jarrah seedlings as a kind of trap plant, I guess, to see what fungi we could find on the roots. And what we found, first of all, was that we had much better seedling growth from those seedlings that were grown in soil taken from inside the enclosures compared to the soil from outside the enclosures. You can see this on the right-hand side, that we had much better seedling growth in the bars on the left, where we had inside the enclosure compared to outside the enclosure. And then we also found that we had different communities of fungi in the seedlings grown in soil from inside the enclosure compared to outside the enclosure. So you can see that in the bottom left. The dark squares are seedlings grown in soil from inside the enclosure, and the open squares are from soil taken from outside the enclosure. So that was really interesting. There was clearly a big difference in the soil fungi between inside and outside the enclosure. And then when we drill down to look at what some of those species were, on the right-hand side, you can see what some of the key species were that we found. And many of those mycorrhizal species we found there were far more inside the fence compared to outside the fence. And as well, some of those other key ecological species like our saprophytes were found much more commonly inside the fence compared to outside the fence. So it's really clear from this work that digging mammals are not only dispersing those fungi throughout the landscape, but their digging action also changes the different types of fungi that we're able to find within the soil, and helps to improve the growth of seedlings associated with that soil. A lot of this work that we've been doing on digging mammals is really showing that those digging mammals have some really clear indirect impacts on soil fungi. So the changes in the soil physical and chemical properties, the increased organic matter, increased soil water infiltration, are all really positive for soil fungi, and as well as the changes in seedling recruitment in relation to those digs, are all really beneficial to the soil fungi and are changing that community of fungi that we're finding in the soil. And as well as that, of course, they have direct impacts in dispersing some of those mycorrhizal species. So what this is really showing us is that it's very important to maintain those diggers throughout the landscape and to reintroduce them wherever is possible, and that this will help to increase soil fungal diversity, increase the tree and ecosystem resistance and resilience. And we really need to think about, there's a lot of work that's going into the control of feral cats and foxes so that we can maintain populations of digging mammals throughout the landscape, not just in fenced reserves where they're primarily found at the moment. Okay, so that feels like a very quick overview of some of the different work that we've been doing, looking at the impact of disturbances on soil fungi and how they might change some of the soil fungal functional groups that are found within the soil. And in particular, I've always been really interested in how those mycorrhizal species might respond to those different changes and how this might lead to changes in ecosystem function and resilience. None of this work ever happens in isolation, so I am extremely thankful to all the people that I get to work with on these projects. All this work taking into consideration the different parts of the ecosystem that we're considering means that I'm often working with lots of other people who have different areas of expertise, and I'm really grateful to be able to do that because I think that gives me a much broader view of the way these ecosystems operate. So I'd like to thank all those people that I work with and all the organisations who sponsor this work, and particularly the students and staff of the Molecular Ecology and Evolution group that I co-lead at ECU, for being such a great bunch of people to work with. Thanks, Jonathan. I think that's it.
That was brilliant. Thank you so much, Anna. We already have one question in the Q&A for you. So David asks, thanks, Anna, really interesting. Did you have a chance to look at fungal succession after disturbance as well as a snapshot in time?
That's a great question, David. Thanks. Yes, we have done that in some other studies. So we've done, we're still working on analysing the results from a study looking at prescribed burning. And that's another one in the Jarrah forest. And we've actually, by some miracle, managed to get some samples from before the burn, as well as immediately after the burn and a couple of years after that. So we're working on that succession now. My PhD student, Aaron Brace, has also been doing some work in the Banksia woodlands, and he's looked at a long-term chronosequence of fire in the Banksia woodlands. So yeah, we're doing that as well. And I think that the snapshot's interesting, but I agree that the long-term succession is much more interesting, and we'll keep collecting samples from these sites and how things go.
Great. If anyone else has questions, please do pop them in the Q&A. If you go to the bottom of your screen, you can click on Q&A. And Nicola has one. Great talk. Is there any knowledge of how changes in mycorrhizal communities might cause changes or shifts in plant communities?
Look, there's a lot of work that in the looking at the relationship between different mycorrhizal species and different elements of the plant communities. I mean, Jonathan's probably a good person to answer this question, actually. But yeah, there are all sorts of specific interactions. So there are some mycorrhizal species, for example, that will only associate with particular tree species. So if you lose those tree species, then you're also likely to lose those mycorrhizal species. There are really clear relationships, for example, between seedlings having particular suites of mycorrhizal fungi and then later on them changing as the seedling gets bigger and older in response to the growth. And so yeah, there's a lot of knowledge in that area, and we can talk about that for quite a long time.
Great. And Teresa has a request that we could actually send out to all of our members. If you could provide like a list of 5 to 10 top papers that you would recommend from your work and your students' work in this area.
Sure, Teresa, I can email you directly if you'd like. Be happy to do that. That'd be great.
So I have a couple questions myself. So you've done work with conifer and pine forests as well. So, you know, thinking about, you know, thinking about this idea of, you know, if you have drought, you're losing some of your ectos, or if you have fire coming through, you're losing some of your ectomycorrhizal fungi. Especially here in Australia, they're so dependent on so few ectos. Have you looked at how, you know, fire is impacting those forests?
The pine forests?
Yeah.
Yeah, so we have just recently done a study where we've collected some soil from pine forests that have been that have undergone a variety of different intensities of fire, and I'm still waiting on those results to come back. But I think that'll be really interesting. And I mean, there's been some great work that's come out of the Northern Hemisphere on the impact of fire on those pine forests. But it's, I suppose, doubly interesting here because they're introduced. And so I'm dying to get my hands on that data and see what it looks like. But I think that'll be really interesting because those specific associations with those introduced trees might show some quite different patterns, I think. We'll see, yeah. I don't know.
No, super cool.
Yeah.
I had an animal related question. So I don't know anything about animals. I'll start right up front with that. But have you actually seen any examples of where these endemic animals that require the fruiting bodies to survive, have they switched over to some introduced ones like tuber or anything like that as a backup? I
believe there is some evidence of that. We haven't found any in our studies, but we have found lots of evidence of, say, quail, for example, adapting to all sorts of different food sources. And this is a fungal example, but you know, they will eat cat food if they find it in the backyard of someone's house. So wow. They can, so, I mean, quenda is quite omnivorous, so they will adapt to a range of different specialist mycophagus species. I'm not quite sure, but I know that there has been some other work done by other people. So I'm not sure about the species we're looking at, like potoroos and woylies, but in other places there has been some work to show that happening.
Yeah, I have one last question, and unless there's another from the audience, we will wrap up with this one. So with your pot study where you're showing that you're able to pull out some of these ectos from scat, I assume you're mixing the scat into the soil. In a natural habitat, is there actually that turnover where the poo is getting, you know, integrated in, or do you think the fungi are actually able to germinate, grow out of the scat and then get down to tree roots?
That's a super good question, Jonathan. I don't know the answer to that specifically, but I do know that most of the scat that we work with breaks down pretty quickly. So we were having one of my students, Beck, is working on potoroo, and they have a lot of trouble collecting scat just in the environment because it doesn't persist very long in the environment. It breaks down quite quickly. So I suspect that it does break down, you know, really quickly and get incorporated into the environment rather than, but it could also be dug in with some of the digging activity of some of the mammals too. But I suspect both probably happen.
Great, cool. Fascinating area. Well, thank you very much, Anna, for that. That was really great today. And We thank everybody as well who attended and everyone who will be viewing this later on YouTube. We'll see you later. Bye for
Thank
now.
you.